field, metabolomics. To introduce Professor Dr. Muhammad Ali Farag, I would now invite Program Manager of comstech, Mr. Haris, to introduce the speaker on behalf of our Coordinator General.
Thank you, Osib. On behalf of Coordinator General comstech, I welcome our speaker for today, Professor Muhammad Ali Farag. He did his PhD at Texas Tech University, USA in 2003. Currently, Professor Farag works as professor at the Chemistry Department, American University in Cairo, where his research work focuses primarily around applying innovative biochemical technologies to help answer complex biological questions in medicine, herbal drugs analysis, and agriculture. Furthermore, Professor Farag works as a visiting professor at the Technical University Munich, Germany, to participate in teaching plant metabolomics and chemometrics modeling for master students. Professor Farag has been recognized with several awards, including Abdul Hamid Choman Award in 2016, Egypt Higher State Incentive Award 2012, and several other awards. For highly cited publications with close to 8,000 citations and an H index of 40, Professor Farag was selected as a top researcher in the field of plant biology in Africa by the American Society of Plant Biology. Professor Farag is also a TWAS Fellow in Agriculture from Egypt. I welcome Professor Farag for today's lecture on Metabolomics Gateway for Milestone Discoveries in the Prehistoric and Post-Genome Era. Professor Farag, over to you.
Okay. Thank you, Professor Harris, and the pleasure is mine to join you again for the comstech. You can hear me well?
Yes, sir.
Great. I will just stop the video because my internet is not that great, and I will share in a moment. And sure, thanks for all the attendees for taking their time on a Friday, special day, to join my talk. I don't have a chat. I can share. You can see it now?
Yes, sir, it's visible.
Great. Full screen. So, welcome from Cairo, Egypt to Islamabad, Pakistan, and to the comstech committee. And here I am giving the talk for today. And again, the title is going to be Metabolomics Gateway for Milestone Discoveries in the Post-Genomic and the Prehistoric Era, as we will see in a few minutes. Before speaking about my metabolomics, I have found this nice video on the internet, and I think it delivered the message on what is metabolomics for those who are not familiar with the science. So let's listen from Mr. Yarmo. You can hear the, 1 second. 1 second, sorry. You can hear the video? No.
We can't hear the sound.
Okay, I will share with sound.
Sure.
So for a few minutes, we will listen to Mr. Yarmo about metabolomics.
This is Yarmo. Yarmo is a Finn, and like a good Finn, he loves trees. He loves them in the forests. He loves them in the xylitol gum he eats after every meal. And he loves them while he is in the sauna smacking himself with Vasta. He loves them so much, in fact, that he has made a career of researching trees. One day, on his rounds of the trees in his laboratory, Yarmo noticed something wrong with some of them. He tried every routine test to figure out what was wrong, but his trees kept getting worse. Jarmo did not know what to do as he watched his beloved trees withering away. Jarmo became depressed, which is relatively rare for a Finn, at least in summer. One day, Antti, Jarmo's colleague, told him about metabolomics, a field of study where researchers can break down samples into their smallest components and assess them in great detail. More specifically, metabolomics is the analysis of cellular processes based on the chemical traces that they have left behind. This analysis then creates a picture of the differences between sick and healthy trees. This can be useful in food production when establishing the chemical makeup of a food or controlling for quality between different rounds of production. It can be used in medical research to help understand the progression of diseases and to assess the impact of a person's diet and metabolism. Jarmo saw that through metabolomics, he could now get a window into what was or was not happening correctly in his trees on the most fundamental cellular level. Now, he might finally be able to figure out what was going wrong in his research.
So now you have listened to Jarmo, and now you will listen to my idea about metabolomics and the research that I do. So stay tuned. Again, myself, I'm Mohammed Farag. I'm the managing editor of this journal. We are all proud of the Journal of Advanced Research. I hope you have heard and you would submit in the future. It's a multidisciplinary journal. And I am a professor at the American University in Cairo and at Cairo University. This is my ResearchGate account, and I specialize in food chemistry and particularly in the quality control analysis of functional using metabolomics. We all know what are functional foods. A typical example is drinking coffee in the morning. We drink it to keep us alert, not to get energy or calorie. So it's for a different function. And this is why we call it a functional food. And it applies for many, many other foods that we eat. But what is metabolomics? I hope Jarmo introduced it a bit, and I will introduce it in my own perspective. Metabolomics started with this picture, actually mostly with the discovery of the DNA sequence by Mr. Watson and Crick. And this happened in the 50s. And the idea was, since identification of gene sequence, that people start to wonder about gene function and what they do in biology. And in 2001 was a remarkable year as well in human mankind, witnessing the completion of the human genome, which was published in Science and Nature. Nevertheless, we are still facing many, many mysteries in science and think that we cannot explain based on gene sequencing alone. This is a photo I took from in Egypt, downtown Cairo. It's an old district called Khan el-Khalili. Maybe one of you have visited. It's a very nice historic area. And what you see in front of you are the people walking in the street, but you can never tell what they are doing in life because they are simply silent. They are not speaking to you like genes. Genes, they don't speak. But when do genes speak? Genes speak to us when they produce their product. And the first product of a gene, of course, is the mRNA, and then the protein, and then the metabolite. And this is the idea that we look at gene products to assess their functions. And this is what we call nowadays in science as biology. And the other picture for this famous ship, Titanic, which sank in the early 1900s. And the reason because of that the ship didn't see except the tip of the iceberg in the ocean, which is exactly what we do when we do science. We don't see the whole picture, but we see what we want to see for the tip, and it smashed. So the idea that looking at gene alone or the genome might not be efficient to tell us about the whole iceberg in science, which is what we call as nowadays system biology. So what we do is that we use metabolomics as an anchor to identify a gene function. Again, the first product of a gene is the mRNA. If it is translated, it's a protein. And if the protein is an enzyme, it gives a molecule. So typically, what we are doing is going against the central dogma of molecular biology and assigning the function of a gene not based on its sequence, but rather based on its product. This was not my idea. It was my advisor idea in 2004. And at that time, it was like this cartoon, what I would call as maybe one of you would see. And I encourage always my students to think the same way. Assigning gene function not based on the sequence, but rather the end product at that time was actually thinking outside the box. So what is metabolomics? Metabolomics is, as I show you this slide, if you happen to be diabetic per se, unfortunately, you will be measuring glucose daily using your sensor at home. And this is what is called a single targeted metabolite analysis. But if you are, sorry, if you are measuring the hundreds, the thousands of chemicals inside your body, this is what we call an omic analysis or a metabolomics. It gives you a better readout of your status, a better idea of the disease progression, and can help you in the prevention or the management. And if we correlate the metabolite level, this is what we call as system biology. Yeah, one would say I see a tree. This is again, maybe for some of you who are acquainted, this is the GC chromatogram from a plant sample. And one would say I can observe 50 or 100 peak, but if we zoom, we can see that we see much, much more. So this is what metabolomics is all about. It's the same analysis you would be doing in your lab, but the difference is the level of detection. And this is typically done on your computer. Omics is not confined only to metabolomics. We often hear about genomics, transcriptomics, proteomics, metabolomics, and now peptidomics. So it's in many, many fields. But why metabolomics in particular of this picture? So this lady eating macaroni, after eating, we observe many changes at the gene level, at the protein level, but most of the changes are observed at the metabolite level. Of course, we don't do metabolomics to monitor the effect of an eating of a certain diet or a macaroni type, but for many other applications. And these are some of them, which I don't do, by the way, but just to give you the global picture. So we use metabolomics to monitor genetic effect, aging as we age, environment, disease, COVID, non-COVID. How does COVID affect our metabolism? Drugs, to identify drug or to predict side effects from a drug. Remember, we are monitoring the metabolic repertoire inside the body. Nutrition, a certain diet, lifestyle. And I think the platform for doing metabolomics is not really complicated. You have a sample, which can be a plant, can be a bacteria, can be a human plant sample. So you do the first step, which is a metabolite extraction, and then you detect your metabolite using spectroscopic technique. And the most common one is mass spec. Then you have the data with you, and the data is simply a matrix of peaks and abundance. And then you identify your peaks. Of course, it's a huge data set, typically in any metabolomics experiment, for which you need to use the multivariate data analysis for exploration. Again, it's a huge data set and wouldn't rumble in the data set as such. And to help you to decide on an answer for your question, we typically use bioinformatics. Bioinformatics I think of as a marriage between computer science and biology. It helps us to get the right information from the data. As I always say, we all have data sets, but maybe we cannot extrapolate the right information from the data. And this is done using what we call the multivariate data analysis. And the most common one is PCA and PELS. So with now, I hope you have understood a bit about metabolomics. And now I will go through applications for better plants and better treatments. Of course, humans are, although I'm not a human person, are more important on Earth, but plants are well deserved as well. So we will see a couple of applications on both sides. I will go through briefly in 4 different areas to give you in brief the application of metabolomics and what metabolomics can add in these fields. So the first one would be, which is actually my area of expertise, because I work in the pharmacognosy department at Carob University, which is including the nutraceuticals and the quality control, disease biomarkers, functional genomics of phytoestrogens, and in agriculture as well. So you can see that the technology is a multidisciplinary and can be applied in different fields. So the first one, which is the phytoestrogen functional genomics, which was my first acquaintance or my first meeting my first date with metabolomics back in 2004 in the lab of Professor Lloyd Sumner. What are phytoestrogens? For those who don't know, phytoestrogens are chemicals. They are produced in only one plant family, which is the legumes. We all eat legumes on a daily basis for their rich protein content. And the reason behind the rich protein content in legume is because actually of these chemicals called the phytoestrogens. What happens is that we know that we all talk in biology that legume plants can make this association with the rhizobacteria in the soil. So if the bacteria sense that this is a legume plant, it come to the plant and establish a relationship with the plant to produce to fix the protein. And the reason behind this association is due to these signaling molecules called the phytoestrogens. And they were called the phytoestrogen because at the human side, they were found to mimic the structure of the female sex hormone and to exhibit an estrogen-like effect. So the question that was in mind at that time was, how do plants really produce these chemicals, or the recipe behind? I call it exactly like a pizza. We all know pizza, we all love pizza. We all know the ingredient, but we cannot make it as good as Italian per se would do, because they have the recipe. The recipe means that not only that I knew I have a flour and the tomato and the cheese, but rather I have the fine details and when was everything added and of what type. So this is the recipe we wanted to unravel. And we were successful. So we monitored, we did it in a plant cell culture, by the way, the cargo plant, and all these chemicals for isoflavones were monitored, identified, and quantified. And we were looking at them as a puzzle, piece by piece, put them together to stack together to build the biosynthetic pathway based on when do we detect them in the cell culture. I'm not claiming the pathway was established long ago, but when we did such an approach, we were able to identify a new pathway, the one highlighted in a yellow box, which was not identified before. And we identified as well the genes that were involved in that. We identified as well, considering that this is a holistic approach for chemical profiling, we identified in the plant a nice chemical called haspidol, and this haspidol showed a very good antifungal activity. And you can see these bioassays, which we done routinely in the lab, where we grow the fungi with the chemical, and we can see that the chemical is restricting the growth of the fungi compared to the solvent control underneath. So we speculated that this chemical is an antifungal chemical, and we wanted to know more, how does the plant produce this haspidol? Going back to the gene expression data, we identified that it's a peroxidase. And as we all know that peroxidase is a very big gene family, and cloning each peroxidase wouldn't be feasible in terms of time or in terms of money as well. So we decided to look at both sides, as I'm telling you, look at the metabolite expression and the gene expression and see which one mirror each other. And when we did that, we monitoring metabolite and gene expression data using the RT-PCR, we narrowed down the gene candidate of the peroxidases from 99 into 3. So instead of cloning these 99 peroxidases from the microarray chip, we cloned only 3. So by that, we have saved not only time, but money as well. We identified the genes that were involved by cloning them in yeast. So this was in brief. I'm no longer doing actually functional genomics. I'm back to Egypt, and the resources are rather limited to do such research. So I decided to focus on something, and this is my message to people who would get trained abroad and then we come back and the resources are much less. So we have to use the less tools, but still we can answer good questions in science. So I decided to focus on nutraceuticals quality control, and I'm not claiming I was the first whatsoever, but I decided to add a different perspective at that time. And this project started in 2008. And this has been, over the years, the question we were answering using metabolomics regarding nutraceuticals or herbal products, the quality control, which chemical makes a health effect, and how does processing affect the quality of the herbal drug. And all of these questions were answered using metabolomics. I give you a glimpse about the challenge of doing a quality control for a herbal sample compared to any other drugs. In case of a herbal sample, we are faced many variables depending on the genotype, the plant came from where, the species, the variety, the cultivar, and people who are among us from the plant would acknowledge such a problem. Where the plant was grown, Pakistan versus Egypt, different habitat. Would it affect the chemical composition? How was it processed, cultivated, or harvested? And typically what we do in phytomedicine or in nutraceutical, we do a targeted analysis. So I give you an example. If you have an apple, another apple, an apple, an apple, and the fourth apple, and you want to choose which one to buy at the grocery store, you would judge by the red color, which is fine. But the apple benefit is not only due to the red color, but rather many, many other peaks. So this is what we are looking at, including all the peaks, all the chemicals inside the apple to judge its quality, and not only just by one marker. And this is what metabolomics would help in that regard. This is my slide I teach to my students to challenge them. So if you were a quality control chemist and you were asked to standardize a tablet containing an apple extract versus the tablet containing aspirin only, which one would be challenging? I would definitely vote for the one on the left, the tablet containing the apple extract, considering the complexity. The typical plant contain hundreds and thousands of chemicals. We don't detect them all, by the way, for sure. So this one is more challenging. This lecture was given in Egypt, so in a different, because we had a Zoom limitation in time, so I had to cut it in 2 parts. We are done with the first part, like a movie. We are in the entr'acte. And give you a tip as a food chemist, of course, when we are in a movie theater, we typically eat popcorn, but I urge you to eat something more healthy, like the apple. And as a food chemist, I give you my tip. Maybe you are not interested in metabolomics, but you are interested in nutrition. Don't drink the juice as we do nowadays. I'm not sure about Pakistan, but in Egypt, people think that drinking a juice is equivalent to eating a fruit or an apple. It's not the case. A juice is mostly sugar, but if you eat the fruit, you get the fibers and the phytonutrient. So eat it and don't squeeze it. And as a pharmacist, I would consider the juice as the generic form of the apple brand. And we all know this quote. Anyone knows it on the chat? You have to vote. Someone to write. An apple a day does what? Can you write it? Exactly. Keeps the doctor away. Good. And we all wish to keep the doctor away. So back to our seats and part 2, metabolomic application in nutraceuticals analysis. It included many plants, and the slide would not accommodate the plants we have investigated. The project was in collaboration with the German side, so we had to please them. We analyzed plant from Egypt and plant from Germany. Dates are Egyptian, hops German, licorice Egyptian, etc. Let me tell you the first story regarding licorice or metabolomics analysis of licorice from across the world. I happened through good connection to collect licorice from different places in the world. And to tell you, people in the Far East, licorice is a famous plant. It has a sweet taste and many health benefits. But in the Far East, they grow the glabra and the uralensis and the inflata in blue and gray. And in Egypt, in the Middle East, and I assume in Pakistan as well, we have the glabra as our famous source. In Europe, they have the echinata. And I simply wanted to answer whether people living in Japan are experiencing a better licorice type than people living in the Middle East. So we collected these different specimens, different genotype, and we analyzed them using metabolomics. The thing that we did differently was doing it comparatively by comparing two different technology platforms. So we compared LCMS, liquid chromatography mass spec, to NMR regarding the analysis of these specimens. And these colors again represent the different genotypes, as I mentioned before. This is called the PCA, and I'm not going to explain PCA much. It's simply a box displaying sample segregation and how they are related to each other. So we can say that the orange is close to the blue and the red, and the green is far away. But what we can see from this slide, importantly, that we don't see the same plot. We see that the LCMS is giving different colors, different circle pattern compared to the NMR. This is that, and it is the same sample and the same extraction method. And the reason behind that when we was be something of importance for using more than one technology per se. And this is what I think of it. Originally, I'm a painter. I take pictures as well, and we all love to take pictures. And the challenge of picturing chemicals in plants or in humans is limited by the detection method. So in mass spec, which has a very high sensitivity level, high resolution, we detect details, and you can certainly tell this picture is for what, but maybe some of you cannot guess. But if we look at an NMR, we see the whole picture, but the resolution is much lower. And till now, have a technology with which we can get this picture as a whole with a high resolution. And this is why we typically use what we call a comparative metabolomics different technology platform. We use NMR a lot as well, and NMR is very famous for natural products for its very strong elucidation power, particularly the 2D NMR experiment. And we use 2D NMR without any separation to analyze hop extract. Hop is used to make beer in Germany and in the Western world, and the plant has many health benefits, by the way. And this is called an HMBC spectra, without going into details. And we can see this minor peak highlighted in the red arrow. We can identify its chemical structure completely, looking at the cross peaks. And we identified at this alcohol methylbutanol, and this is the sedative principle in hops. And we identified doing this analysis by that this chemical, which is the sedative hypnotic in hops, is not a real chemical. It comes after the degradation of the active constituent in hops. And this is opposite to what we would say, preserve the drug to keep the activity. But here was an example in which we would degrade the chemical agent in the plant to get this agent if you are targeting a hypnotic action. And was published. And this was how I started to get to the prehistoric era. Maybe some of you would wonder, why did he choose such a title in post-genomic, as I explained it before, and prehistoric era. Analyzing hops as a source of beer made me get acquainted with a different study that was in Egypt about 3 or 4 years ago in Upper Egypt, and we all know that the historic monuments in Egypt are in the Upper Egypt. They discovered what they believed at that time by a Japanese mission, the oldest brewery and biggest brewery in mankind. And beer was a commodity in old Egypt, by the way. And these are the vessels where they cook the beer. And to the left are the vessels where they fill the beer and sell it. Let me show you where the location is. If you visited Egypt one day, we would love. It's a very nice country if you haven't yet. And this is the map of the site. So this is Egypt, and it is on the West Bank in the Upper Egypt area, close to the famous city of Aswan. It's called Heracleopolis, by the way, and this is the side, the aerial site, and these are the vessels where the beer was cooked. And the design told us that it include a firing step, and it is likely to be a design for a beer factory. When we analyzed the C14 data, to date this beer factory, it was 4000 BC. It's one of the oldest, but not only the oldest, but the biggest. When we calculated the volume of the factory, these vessels would produce on a daily basis 650 bottles of beer, which is a big scale at that time. So why I would be included, this is the picture of the Japanese scientist, Dr. Baba, who worked on the excavation. And they wanted a clue from a chemistry perspective that this is beer and the recipe. So they cannot export these things abroad because it's a monument artifact. So they had to struggle with us in Egypt to help them in that regard. So to identify the chemical makeup of the beer inside these vessels, again, remember it's a pizza, a recipe of the pizza, but a pharaonic pizza dating 5000 BC. And I took the challenge. So the first thing that I collected the residues and we tried analyzing them. And one of the techniques we used was the GCMS. And what we identified actually that the ancient Egyptians knew about flavors. We can see that we analyze from vessels on the same line, they have a different profile from other lines. And this is exactly similar to nowadays what we call in a factory, a production line. And this tells us that the Egyptians knew about flavors and different chemical composition. But that was not the only discovery. The biggest discovery was that the active agent, the preservative that was used in these vessels was something that is still present in our Coke, which is phosphoric acid. We cannot claim that the old really, because phosphoric acid was identified by the Romans, didn't find the vessel with that it were from a natural likely where they used to brew. And we happen to solve the recipe of the Egyptian beer as well. So this is why I was called metabolomics can be not only a gateway for discoveries in the post-genomic era, but the prehistoric as well, about ancient drugs, the ancient cosmetics, and storing the ancient foods. And I have another lecture, maybe one day. It's a long lecture, which I call an ancient Egyptian food and practice between myth and science using metabolomics.
I think Professor Farag accidentally logged out for some internet issue. Let's just give it a minute for him to log back on.
You can hear me now?
Yes, sir.
I'm sorry, I got disconnected.
No.
Share.
Yes, the screen is visible now.
Great. So this is a talk maybe we can think about in the future about ancient Egyptian food and between myth and science. So again, back to nutraceuticals, and questions that we have been answering using metabolomics is by looking at processing method, cold versus hot. This is the plant. I'm not so sure do you have in Pakistan or not. We drink a lot in Ramadan time, particularly called Roselle or hibiscus. And Roselle has a very nice, distinct flavor. It has a red color. And typically, there is a myth about Roselle. Roselle, people use to lower their blood pressure, and they believe that when it is being cold, it's better. It's lower the blood pressure. When hot, it increased the blood pressure, opposite action. So we decided to answer such a myth and whether it's real or not. We prepared Roselle using different extraction method, hot versus cold, decoction and infusion are hot preparation method, and cold maceration as a cold one. And we looked at the chemical profile using NMR. What we identified that when we extract it with a hot solvent, we extract more of the slimming agent called the hydroxycitric acid. And this is why when you want to use it to lose weight, it's better to be cooked at a hot temperature. But if you want to recover more of the anthocyanin behind the antihypertensive effect, you better extract using a cold method. It's a simple experiment, so we can see that different processing method can have a different function at the end. And the work was published in Food Chemistry a couple of years ago, work of my PhD student at that time, Ms. Dalia Rashid. Carob is also another example for a plant in Egypt, which is grown in the Mediterranean region, the work of my student, Dr. Dina El-Ersh. And carob is famous for its sugary taste. We were looking, we published a lot on carob, looking at the effect of origin and roasting effect, because carob has to be roasted before its inclusion in food. This is the carob tree, if you haven't seen. It's very famous in the Mediterranean region. It can be eaten unripe, and it can be used as antidiarrheal. As it ripens and it gets brown in color, it gets more sweet, as typically as any fruit. And in Cyprus, they have the chocolate carob and the syrup carob. And it's a very good source of this sugar called the pinitol, an anti-diabetic sugar. Simply what you will answer are three questions. How does the geographical origin affect carob composition? How does the ripening stage affect the chemistry of the fruit and the roasting effect? So these questions were all answered using metabolomics and bioinformatics. I collected samples only from Egypt, from 3 different areas, Giza and Alexandria and Siwa. Siwa is a very remote oasis. Neighboring Libya Valley is a very different area where you have to travel 12 hours until you reach a green zone. It was visited by Alexander the Great 5,000 BC years ago, and I visited as well in 2018 when I collected the samples. So we were looking at ripening effect, roasting, and origin effect with carob. Using GCMS, what we identify that as the fruit ripen, sugar profile is changing from maltose, which is not that sweet, into sucrose, which is more sweet. And this explains the better taste of the ripened fruit. But regarding the phenolics, we think that the unripened present a better source of the tannins compared to the ripe and the roasted fruits. Metabolomics and drug discovery, and this is the dilemma of all people working in natural products, finding the needle in a haystack and identifying the active agent behind the chemical effect. And an example is caffeine in coffee. We all know that for sure, but in other examples, we don't know the chemical behind the efficacy of a drug. And the plant is coming from Aswan again in Upper Egypt. And Aswan is very, very famous for the scenic scene of the Nile and the date market. They have a huge date market where you can buy all different types of dates. Dates are common in the Muslim world and we eat them a lot due to their rich sugar content after fasting. But people who are diabetic are not allowed to eat dates because of the high glucose level. So this date, which is grown only in Aswan, called balanitis, is the opposite. So it typically lowers the blood sugar against other date fruits. So I took and to identify the active agent, I decided that to help me figure out, I decided to compare it to other date types. So I compared it to the sweet dates, the regular one that we eat at home. Using NMR, we can see that they are loaded with sugar signals, but looking on the aromatic ring in the yellow bar, we can see signal present in the balan, absent from the sweet date. Doing further and doing the 2D NMR experiment without addition, we confirmed that the anti-diabetic balanitis date contained this famous chemical called trigonelline. And trigonelline is very well recognized for its anti-diabetic action. And example, like when you identify aspirin in a plant, you say that the plant is an analgesic, and you don't need to do a biological assay because aspirin is well recognized. Similar way, and there was a project from the European Union to a company, 1 million, and they couldn't identify it for many reasons. The plant is the compound is charged and can escape isolation attempts, but in one experiment we managed to identify, and the level was really abundant in this plant of the trigonelline. We also try to explain things in medicine, and this is something not in Egypt, but in Eastern Europe. We all knew bee and honey, and we know how honey is made. The bee land on a flower, they get the nectar, and then convert it into the sugar, from sucrose to glucose, fructose, and then it's a honey, gooey honey. But another function for the beehive as a source of medicine, interestingly, is more famous in Eastern Europe, where people would go to a clinic and smell the beehive air, believing that this bee air can relieve many diseases, especially bronchitis. So what you see here are ladies, they are not sitting in a sauna, but rather sitting under hive underneath. And we wanted to identify what would be in the bee air behind that. So this young girl, instead of smelling the beehive air, we can get a chemical and put it in an atomizer as a better way. We are still working on that, and we do some good results proving that the beehive air have antimicrobial effect against several lung pathogens, and we are hoping to identify the active agent by time. This is my student Ahmed Al-Banna, and I'm not against isolation. We all do it as natural product chemists, but sometimes when we do isolation to identify the active agent, we lose hope at the end, and we cannot reach our goal. And maybe metabolomics, and this is something related to Egypt, because we all knew a couple of centuries ago, if we wanted to travel from Egypt to Pakistan, we would have taken this route. But nowadays, with the aid of the Suez Canal as a shortcut, we can travel in much short time. And this can be equivalent to metabolomics as a technology to help expedite natural product drug discovery. Sometimes you go for the bad things as well to analyze, not only the good ones. And this is Hoka. I'm not so sure if it's famous in Pakistan or not. It's getting to be a trend in Egypt, and people think it's more healthy than cigarette smoking. And it comes in many, many different flavors: apple, orange, melon, grapes, etc. And I decided to compare them and compare the aroma compound. They are not dangerous, they are okay, but they are very bad, not because of the flavor, but rather to the coal tar that is included to help them burn. And I think that it produces many pro-carcinogen compounds, and it can be even more dangerous than cigarette smoking. The disease biomarker I would skip away for the sake of time, but again, metabolomics, and this is actually the rigor of the technology is more in the medicine side to identify markers for disease. I happened to be included in one study regarding heart diseases. I will skip and I will go to the last one, which is the agriculture aspect. Sorry. Yes. Why do, when we have a food at home, which is spoiled, it's due to, we smell it and we know that it has been spoiled because of the bad smell of the food. So why does bacteria or fungi release these aroma compounds, the bad smell? This is something interesting that happened to have an agricultural application. I would let you take a minute and look at the slide. What you have in front of you, pay attention, is a plant, and the plant is grown on one side of a plate, and a bacteria is grown on the other side, on the left, on the right. On the left, the plant is on its own. There is no neighbor beside. And what you can see that the plant neighboring the bacteria is growing in size much bigger. But bear in mind, there is a wall in between. So there is no physical contact between the plant and the bacteria. So how come that these bacteria are inducing a cross promotion effect? If anyone can think of a simple answer to this observation and put it on the chat, I'd be happy to see. Think for a minute. Yeah, so what we have here is a plant growing on one side. Bacteria may release useful nutrients. Dear Lubna, there is no way that the nutrient would reach through the Petri dish. There is a wall in between. The media is the same in both cases. It's the same media. Signals getting close, but what type of signals exactly? Photosynthesis, exactly, aroma compounds. I congratulate the lady or the sign. I wish to read Hassan. Congratulations, Hassan. This was right. What's happening here that the bacteria is releasing an aroma compound, a perfume, and the plant smell and respond to an increase in size. And this is what we were looking for. So to prove the processes, we have to identify this volatile profile and identify the active agent. So we grow these bacteria in culture and collect the aroma compound using an SPME fiber. And the SPME would absorb the volatile compound and then analyze them using. This was protocol. We were really lucky at that time to identify the active agent in the bacteria, and the protocol for collection was published a few years ago in Nature Protocol and was actually featured on the cover page. So, and I call it but the publication, and I don't do it because I'm really not good enough doing the application side. But the publication is not the application. It's like a fruit, you eat it unripe, but if you get it to the society and apply it, you enjoy the full taste and the flavor. And one application would be to apply it in agriculture and to take this chemical spray on the plant to induce a growth promotion effect. We are now interested as well in human gut microbiota, which is a hot area in science. We all know that the humans, we are in our intestine, we have millions of bacteria, and they play a function in our body. It's a dark side that hasn't been explored, and we are using metabolomics to explore. So in your gut flora, you have the good bacteria and the bad bacteria. The good bacteria can flourish when you eat fruits and fibers. And when you don't eat these and you eat more of the processed food, taking antibiotics, the bad bacteria becomes more dominant. These are the function of the gut microbiota in immunity, in vitamin biosensors, in mucus secretion. And the question now we are asking that many people knew the effect of gut microbiota on nutraceuticals and how the gut microbiota affect the functional food chemistry, but no one has really looked at a different perspective, which is how does, when we drink our nutraceuticals, how does it affect our bacterial metabolism? When you drink your coffee in the morning, does the bacteria like that or dislike that? And this hasn't been really addressed before. So we did it by examining different functional foods, coffee, tea, pomegranate, and putting them in culture with the gut culture. It was an in vitro assay, of course. And we profiled the bacterial metabolites, CMS. We identified how does the bacteria respond to these functional foods, typically by increasing the short-chain fatty acid. But one very interesting finding that we identified that these functional food increase the level of the GABA, the gamma aminobutyric acid, and whether this increase in the gut can be sensed in the CNS, is it possible or not? And we all know that there is the famous gut-brain axis in our body. It was published in our favorite journal, Journal of Advanced Research, and I think it was a good study because it's heavily cited till now. We are also interested in looking at the balance and how do good chemical interact with bad chemical in our gut. It's not always peace. We had contaminant in our food, like these polyaromatic hydrocarbon. We have them when we over-grill the meat, and they are not a good chemical. And so we take both the good chemical and the bad chemical and put them with the gut microbiota and see what would the gut microbiota do in such a scenario. With and without, realize using what to identify that actually the gut microbiota can quench the bad chemical and prevent oxidation to propagate, which was not happening when we don't have bacteria. This is the work that we just published this year, the work of my not student, my collaborator, Dr. Zainab Abdushakur at Natker in Egypt. And I think it is one of the first papers identifying the role of gut microbiota biotransformation of procarcinogen in the presence of antioxidant. Let me share with you one good news. I am hosting this year a special issue in the journal Food Chemistry called Metabolomics in Food. It has just started, and this month is the first month till the end of the year. And if any of you are interested, you can contact myself or my co-editor, Dr. Milan Georgiev. With that, I would like to thank you all for your time today, and it's a Friday, blessed Friday for all, and Happy New Year. But let me thank the people who contributed in my career at the Leibniz Institute for Plant Biochemistry, Dr. Porcel, my predecessor in NMR. We did a lot of work on corals, didn't have time to talk about. South Korean Institute for Science, the work we did in with the bacteria and the plants, the gross promotion, the work of Professor Chongmin in Korea, and people who have really helped me in my career, my advisor, Professor Paul Pelé, Dr. Porcel in NMR in Germany, and the family in Egypt for enduring all the travel. And with that, as a food chemist, I bid you goodbye and thank you. Thank you for listening to Yarmo and myself. And I hope it was not too much. I know the slides were really, I wanted to deliver all what I have. I hope it was not a heavy meal. And I think this is something that before the lecture lesson from being in academia. And I'm open for questions, if any, for 10 minutes.
Thank you so much, sir. So to all the participants, please write down your questions in the chat so Professor Farag can answer them.
If they can, by voice, I'm fine as well. Okay, let me. Thank you, dear. Anyhow, if you have any questions, anything in mind, you can reach me by email or on ResearchGate as well. And looking forward to your questions, if any.
They can also send the questions to me as well, and I'll compile them and send them to you. So we'll just give it a couple of minutes and see if anyone has any questions.
Okay.
Otherwise, I'll just take them over email, and any questions, I'll forward them to you to answer.
I know in Pakistan you have this excellent center by Dr. Attaur Rahman.
Yes.
Which city is it? Which city? Now led by
Karachi.
Karachi. Okay. Yeah.
Yes.
It's
You mean
one of the
ICCBS,
best.
right?
Yes, exactly. Yes. Yeah.
Our coordinator
Dr.
general
Chaudhary.
is also the director there.
Yeah. Dr. Chaudhary is now the director, if I'm not right.
Yes, he's also the coordinator general of comstech.
Okay. Well, if you happen to be in contact with them, send them. I don't know him and I never met him really, but he is highly respected and my regards.
He was meant to be present here for an address, but last minute meetings. So that's why.
No, that is understandable.
But I've passed on all the information to him, your lectures.
Thank you, dear.
Okay, we have some questions
can
now.
Develop, yes, if pathogen can develop drug resistance to antibiotic, do you think the possible food can be modulated to get desired nutraceutical? Well, it's we all know. Yeah, it has been a long field now that we all know about the antibiotic resistance against antibiotic and the hope of natural product to be another source. Whether or not the problem in gut metabolism, we have to remember that these chemicals are typically detected at small levels, and to do the bioassay is not very convenient. You need to scale up the cultures, and the efficiency of the biotransformation is not that high. This is my experience, maybe not quite true, but this is what I have seen. But there is hope, of course, and all these, and of course, remember that it's done anaerobically. It's not very easy to maintain, not any lab. It's not like any bacterial culture, as you would know, but it's an area that hasn't been explored yet. How can management of antimicrobial resistance, can you also work on antiviral? I don't do antiviral at all, and not my, no, I have never worked with viruses. But viruses as well, remember, I think that viruses wouldn't interest me because the virus, they don't produce chemicals. Yeah, it's just a genetic material and a protein, maybe. And this is why I didn't get to work on COVID as a challenge. Chemical parameters can be used to judge the quality of eggs, apart from the physical property like weight, albumin, yolk. Well, I'm not acquainted with all fields, but of course, I have thought of it before, and I wanted to do a metabolomic study on comparing different egg types, especially organic ones. And of course, what we all eat at home are not organic eggs, as we all know. They are commercial eggs. But egg is also is rich in cholesterol and many sterols, vitamin E, and many other phytonutrients. It's a complex mixture. It's not, but it needs someone, which not myself, because it needs someone expertise in proteomics and proteins, and I don't do that. I don't have I don't have any experience in proteomics. Many natural products, is proteomics more helpful or metabolomics in detecting a disease? It really depends, my friend. You have to go for both. Metabolomics is surely cheaper and more simple to develop here in Egypt. Proteomics is not as is more expensive than metabolomics from my reading, but both has to be considered. But it's just troponin per se. The famous marker is a protein. Is not a chemical. It depends. Many natural products is very sensitive. If we isolate it, how can stable it? Yes, of course, this is something else we all know. So the natural product in the original source is stable on its own, sustained by the matrix itself, like vitamin C. Once you isolate, you have to be very depending, of course, when you see that so once you isolate at first and you do an HPLC and then you leave it and then you find that you see other peaks, it's a sense that the chemical is unstable. And the best way is to store it is no oxygen and at low temperature, and avoid moisture as well, of course. Please do more sessions in future too. I think I've done now enough for Comstech. This is 2 lectures, so I think you need to take a break from me and maybe promise in the future if I have something new, I promise to share. Inshallah. First of all, thank you so much. I want to know which metabolomic technique is best for analysis. Bacteria. I think that especially in our world, in the developing, GCMS is good enough, I would say, if you're targeting primary metabolites in bacteria, but if you are seeking secondary metabolites, then you need to revert to LCMS or UPLCMS, I would think. Again, there is not one technology with which you can say, I'm taking the picture of all metabolism. It's always cons and pros, and you have to use more than one. And this helps as well to solidify your study and your conclusion. Phytochemical analysis best technique is HPLC or GC. This is Samia is not fair questions. We all know GC is more suited for volatiles, essential oils, aroma compounds, the best of the best. Like this bacterial question, it wouldn't have been solved using mass spec, LCMS or HPLC. But bear in mind that most of the phytochemicals are polar in nature and they don't fly, as I teach my student. I have a nice slide on that. So for that, you have to use the HPLC for analysis. It depends. I like the way you give the lecture in form of story to connect ideas. Yes, appreciate it, Lubna. It's my pleasure. I like that as well. I used to be a painter, tell you the truth, and painting made me had this idea that you have to simplify everything as a painting so people can understand. And I think this is inspired from the artistic side that I have. I don't do great science. My science is normal, but I enjoy putting the artistic side to the science. And you should do. When you do it in your lecture, the student would be much more engaged as well, by the way. My advice to you. I really enjoyed it, and I wish you all a blessed, happy Friday all over the world. And thanks to the Comstech, Dr. Hasib, and all the people who organized it. And I'm thankful that it's a Friday and I see this big participant. I'm giving the same talk, by the way, if you wish, or you have missed it, but on a different Zoom link to China on January the 10th at 10 a.m. Cairo time. If anyone would like, I can send him the Zoom link for this session. It's exactly the same lecture, by the way. So with that, I say Assalamu alaikum and have a great day.
Thank you so much, Professor Farag, once again for the lecture. And thanks to all the participants. We have had a good participation today. Even though it's a Friday, like you said, we have had around 140 people joined in. All thanks to you because of your brilliant way of teaching. It has been a very interactive session. So thank you, Professor Farag, once again, and thank you to all the participants. We have had 2 virtual sessions, so hopefully in the future we can also arrange a physical event for you to travel to Pakistan. And thank you, everyone. And with that, I will conclude the webinar for today. Thank you all.
Thank you all.